Good morning, everyone. Welcome to week number 5 in Biology 120. We're past our halfway point now in the course, so let's see what we have done last week and what we're going to be doing this week. Oh, can't find my cursor. Anyway, there we go. So last week you learned about metabolism, which is all the biochemistry that happens in cells, breaking down food, making molecules that you need. And this included a discussion of cellular respiration, which is a way that oxygen is used to make ATP molecules from food. But we also covered anaerobic respiration and fermentation, ways that ATP is produced without the use of oxygen. You also learned about photosynthesis, which is a way that some organisms on the planet use sunlight energy to combine carbon dioxide and water molecules to make the sugar glucose, which is the food source for these particular organisms. So that's what you learned about last week. This week is going to be all about DNA and the main way that cell division takes place. So read your chapters 10, 14, and 15 in your OpenStax textbook. There's some good videos in that chapter too, so take a look at those. Chapter 10 is all about mitosis and cell division, normal cell division. So take a look at that. But also chapter 14 is about the DNA molecule, which of course is awesome, awesome, awesome. There is mine back there. And how DNA replicates or makes a copy of itself, which we're going to talk about in just a second with my little diagram there. And then chapter 15 is how DNA is actually used, because yes, you know DNA is the genetic code in cells, but then what is it used for exactly? It's used to make proteins. That's its most important job. It makes all the proteins that a cell needs. And what are most of those proteins? Enzymes. And enzymes run the biochemistry of cells and give them their identity and do the work that's necessary for a cell to survive. So that's chapter 15. Read the Module 5 overview and watch the videos. There's some good videos, and there's a definitely pretty good one on cancer. So this week you've got discussion board postings that are due and milestone 3. All right, so let's talk about your discussion board postings and the questions that are posed to you. One of the questions is cancer a disease of mitosis, which really means cell division, or a genetic disease, which means a disease of genes, or both? And like I mentioned last week, if anybody asks you a question like this, the answer is always going to be both. Otherwise, they would have not tagged on or both. So let's talk about that for a second. So what is cancer anyway? Cancer is your cells that are dividing out of control and divide and divide and divide. And the most important property of cancer cells that makes them so dangerous is the fact that they metastasize. And that means that they move throughout the body to other locations from the primary location. And that is not good. Okay, so let's get back to mitosis or genetic. Let's take a look at the cell cycle here for a second. I made this little diagram on the right-hand top corner here. And there are representations of this in your OpenStax textbook and some of the videos that you're going to watch. But I want to emphasize a couple of things. So you start at 12 o'clock, right? That's the top of the cell cycle. So what is the cell cycle? It's a description of the life cycle of a cell. So we start around to the right-hand side. We're looking at this clockwise. We have the G0 phase. Okay, but G0 is part of G1. So let's talk about G1 first. G1 stands for the growth phase. This is where the cell increases in size. It increases the number of molecules that are made in it. It increases the number of ribosomes, mitochondria, chloroplast. It increases its organelles. Everything that it's going to need in order to divide. All right, so that's a general growth phase of the cell. That's G1. I'll get back to G0 in just a second. Then we have the S phase. S stands for synthesis. And this means that the DNA is synthesized. in this phase of the cell cycle. The DNA is replicated. You have to make a copy of the DNA to end up in the 2 new daughter cells in order to have the right genetic component in each. So that's what happens in the S phase. And then we have the G2 phase of the cell cycle. This is where we have all the cellular machinery that is produced or replicated, it depends on what they are, in the cell that are needed for cell division. So the centrioles, the mitotic spindle, which includes the spindle fibers, etc. So all that machinery that's necessary for cell division gets replicated or produced in the G2 phase. And then we have M and C up at the top. M stands for either mitosis or meiosis, and that's whatever type of nuclear division, including division of the chromosomes, happens. That's the M. And then C is cytokinesis. That's where the rest of the cell divides in half. So the cytoplasm divides into the 2 new daughter cells. The plasma membrane does. The cell wall, if your organism has a cell wall, but of course we're human beings and we don't have cell walls. Okay, so that's what happens in the life cycle of a cell. Now let me go back to G0. So during the G1 phase, the cell will reach a particular size and have a number of organelles, the right number of organelles and other molecules in the cell, where it can actually do its work. So in G1, there's a stopping place called G0. This is where the cell actually does its work. So every cell, if you think about every cell in our body has a particular job to do. So for example, a heart muscle cell. The job of a heart muscle cell is not to make new heart muscle cells. It is to contract and relax, and that's to make your heart pump so that your blood flows through your body and oxygen is carried throughout the body to the different tissues. So the heart muscle cell has a job to do, and that is to relax and contract. That's what it does in G0. So this stopping place is where the cell does its work. It's a very important part of the cell cycle because cells have jobs to do. They don't just hang around to reproduce, okay? Now with cancer cells, they don't stop in G0. I also didn't mention, but there are checkpoints all along these phases. Checkpoints are just stopping places where the cell checks itself and makes sure everything's in order before it moves on to the next phase. Those are checkpoints. Cancer cells don't ever stop in G0, and they ignore all the checkpoints in the cell cycle. So cancer cells divide and divide and divide and divide, and that's all they do. Just constant cell division in your bodies. The other problem with cancer cells is that they metastasize, and that means that they move to other locations from the primary tumor site and create problems in other tissues. Okay, so that talks about mitosis and cell division. Is cancer a problem with cell division? Absolutely. Is it a problem with genes? Absolutely. You have heard of some of these genes before. If you've ever heard of the BRCA genes 1 and 2, put you at much higher risk for developing breast cancer and or ovarian cancer. So you know that there are cancer genes because you've heard of them before. So look up more information about those so that you can write about that topic in your discussion board posts. You're also going to talk about how lifestyle changes can lower your cancer risk. Nothing lowers it to 0 because you're getting older every day. You're taking on more environmental damage every day of your life, every day that you live. Plus, mutations just spontaneously happen in cells. So there is absolutely nothing that you can do to make your cancer risk be 0. But there are lifestyle changes that you can make to lower your cancer risk. You're also going to talk about in your response post environmental carcinogens. A carcinogen is any type of mutagen that produces cancer. And there are plenty of environmental carcinogens out there so many that we breathe in, that we eat on our food, that we through beauty products. Oh my goodness, there's just so many. There's so many out there. And how you can remove these from the environment. Okay, so that will be important too. And then you've got your milestone 3 of your final project. And this is all about ways to solve the problem that is your particular topic. How are we going to go about fixing this so that this is not a problem in the future? So, for example, global warming. How are we going to do this? Well, we've got to stop burning fossil fuels, period, because that puts too much carbon dioxide into the atmosphere. That's what we're going to do. How are we going to do it? You're going to explain that. Ethical consideration. This is an extremely important part of your final project. You need to find out what are the laws, what are the regulations, what are the guidelines that we already have in place to help solve this problem. If you're talking about endangered species, one thing that you absolutely have to mention is the Endangered Species Act that we have. But are there also state laws? Are there state guidelines or regulations? For example, hunting regulations. Hunting regulations have to do with endangered species. So look those up because they're important. These are laws and guidelines and recommendations and regulations that help your particular topic. So do discuss those positive and negative aspects of the issue. You know, there must be a reason that we just keep on burning fossil fuels. And that's because there are several industries that are in business because of this, right? So that's an issue. And personal impact and how you can make a difference. You as one person can make a difference with this particular topic. So that's all milestone 3 of the final project. Now, what's interesting here is that you're going to change the format of your particular milestone. This will be PowerPoint slides. And remember, I said, you know, they don't have to be PowerPoint, but they have to be slides. So Google Slides or whatever. You're going to make slides this time and make sure you label all the sections. One slide is going to say methods of conservation. Another one, education. Another one, solutions. Okay, so label these sections. Include citations in text because you are going to look up information about your topic. And include at least 1 reference slide because you should have at least 3 references for this particular section. Okay, and then I'm going to add a little bit extra this week because since this is our DNA week, I do want to cover a little bit about DNA fingerprinting. You can stop here if you want to, or you can listen on because this is one of my favorite topics. I did my research when I was in graduate school on the technique of DNA fingerprinting. I used it later on in my research, so you know, this is one of my favorite topics. So DNA fingerprinting is also called DNA profiling, typing. It's got lots of names. So let's talk about it and how it is used. In 1985, the picture of the man down there, Sir Alec Jeffreys, he was not knighted at that time, so he was Dr. Alec Jeffreys, developed a technique. He was a geneticist, and he developed a technique that he noticed he could tell the difference between people and samples that he had because of this particular technique that he had developed. So he went home and he told his wife about this technique that he was using, and he could actually figure out who were parents, children, etc. And he said, this is pretty cool. You know, you can look at DNA and you can figure out whose DNA this is. And she said, that sounds like fingerprinting. And lo and behold, that's where we came up with this name, DNA fingerprinting. It's got nothing to do with fingerprinting. It's just a way to look at DNA and figure out whose DNA that is. So in 1985, Sir Alec Jeffreys had developed this technique, and Britain's police departments had been reading about this And they had some cases. They had 3 cases of teenage girls who had been raped and murdered. And they had no idea who had committed these crimes. And so they went to him and they said, listen, could we use this technique of yours to figure out who killed these 3 girls? And he said, sounds good to me. Oh, let's try it. And so they started collecting blood samples from all of the men. We could never do that in this country. From all of the men in the 3 villages where these crimes happened. And they started sending all the blood samples in to Alec Jeffries. 5,500 blood samples were sent to him. And of course, back then, this particular technique took a long time. So he was testing and testing and testing and testing. And then they got a confession from someone. So they took this person's blood and sent it off to Alec Jeffries, and they said, okay, we have a confession, full-blown confession from this dude. Run his DNA and see if it matches the 3 samples of DNA you had from the rape cases. And Alec Jeffries said, good news and bad news. The 3 DNA samples that were crime scene evidence all match. However, the sample that you sent me does not match. And so the person who confessed did not commit these crimes. So they went, oh man. So the first case of DNA fingerprinting that was used in criminal justice exonerated an innocent person, which is kind of interesting. But the police got wind that someone's name was Colin Pitchfork, had gotten one of his friends to go in and give blood for him. Well, the friend, Ian Kelly, went to the pub later that night and was laughing and joking with all his buddies, and he said, oh yeah, man, my friend Colin Pitchfork asked me to go give blood a second time, so I did. And one of the women who was there, the hair, pulled up on the back of her neck. And she said, ooh, this does not sound good. So she went to the police and told them about Colin Pitchfork. And they got a blood sample from him, and sure enough, he was the perpetrator of these 3 crimes. Now, he was not a suspect in any way. He didn't know these 3 girls. He was fully employed. He owned a house. He had a wife, children. No one suspected him. And he did these 3 crimes. So he was arrested and now is in jail in Britain and will stay there until he passes away. In 1988, the FBI began using this technique. So really, it hasn't been that long since we started using DNA. The first technique that was developed was called RFLP, Restriction Fragment Length Polymorphism, which, okay. And it produced a DNA pattern like the one you see on the right there. That's what Alec Jeffries developed. Well, then we figured out a way to use PCR. And you guys have heard of PCR lately because it is one of the ways that COVID is tested for. And PCR multiplies any genetic information that's there that can then be detected. So PCR is the new type of DNA fingerprinting technique where we can take the most minute samples of DNA that could be present on a cigarette butt, on a straw. Somebody blew their nose in a Kleenex. Now there's touch DNA. You can just swab door handles and find DNA. That's using PCR. And there are even newer techniques that are being developed where eventually DNA fingerprinting will be done out in the field. So that's very exciting. There are lots of different techniques now that come under this umbrella of DNA fingerprinting. The newest things that have been happening have to do with genetic genealogy websites and investigators. So DNA from crime scenes have been turned into different DNA websites, and we have used that information also along with family tree analysis, so that's genealogy, to help catch criminals. One of the most famous cases in the last few years has been the Golden State Killer, who went undetected for 40 years. His name is Joseph D'Angelo, and he was caught through this particular technique. So the crime scene evidence was turned into a website. The family tree and possible relatives were determined, and then through the analysis of the family tree, they narrowed it down to possible suspects. They got a sample of Joseph D'Angelo's DNA without him knowing it, and sure enough, he matched the crime scene from 40 years ago. So this is a new exciting way that DNA is being used so that criminals from a long time ago can actually be caught now. So that's DNA fingerprinting and how it's used in criminal justice. It's very interesting if you love criminal justice, true crime, and all that. This is an exciting new development since the 1980s. And it's one of my passions, as you can tell. If you have any questions or cases or anything you want to ask me about, please just email me. Also, if you have any issues this week, please let me know. m.sigmon@snhu.edu. I will be glad to answer questions for you. So with that being said, I hope that you have a wonderful week this week.